Good morning, class. My name is Doki Simon James. Today we shall be looking at the excretory organs. Uh, First of all, what are the excretory organs? In the past slide, we shall be looking at uh, some few of these particular body organs that are concerned with the role of excretion of waste products. We start with the kidney. We say the kidneys are responsible for the excretion of water and electrolytes to maintain the normal volume and composition of the body fluids. The next one is the skin. What does the skin excrete? The skin contains sweat glands that are responsible for the excretion of small amounts of nitrogenous compounds, okay, water, and some electrolytes. This one forms the component of the sweat, sphenoid. We also look at the lungs. The functions of the lungs is to eliminate carbon dioxide and water, and in a case scenario of impaired lung function, there will be retention of carbon dioxide and what we call acidosis. The intestines excretes the digestive waste. So, in this today's lecture, we shall be looking mostly at the urinary system as a whole. So, the diagram we see here is an illustration to show the location of the urinary system, right from the kidneys, then the ureters, the urinary bladder, and then the urethra. All this forms the component of the urinary system. And we shall be looking at each of these particular organs and their physiological functions one by one in the upcoming slides. So the kidneys bilaterally located, okay. We have two, one at each side, left and right. The ureters, of course, the ureter is the one connecting the kidney to the urinary bladder. So we also have two of both sides, left and right sides of the body. And then the urethra is one. This is the exit for the urine. Okay. We said the kidneys are protected by the lower rib cage. Only the kidney makes urine, and the urethra carries the urine to the urinary bladder, which temporarily stores it. And when the bladder contracts, the urethra carries the urine to the outside of the body. So this summarizes basically uh, the anatomical and also physiological functions of the renal system from the kidney up to the urethra. On the right here, we can see a cross section of the kidney going from outside inwards. You see the outermost part here uh, is what we call the cortex. Okay, the cortex of the kidney, and then going in, we have the medulla. The medulla is this particular kind of layers here, and then in between, we have the renal column separating the medulla. We have the renal pyramid, which is part of the medulla, and then we have the calyx. This is a point where the, the medulla empties into the collecting tubules. On the other side, we are looking at the blood supply of the kidney. We have the renal artery, the one in red, okay, and normally arteries, veins follows arteries. So alongside it also we have the renal vein, okay, and this part here where the ureter connects together with the with the part of the kidney is what we call the hilus. So this is basically a brief overview of the transverse section of the kidney. So let's look at the first briefly. We have the renal cortex, renal columns, renal medulla, the pyramids, and then the renal pelvis. The renal columns and the renal pyramids are concerned with urine formation, and the urine drains into the calyxes, and then into the renal pelvis, and then to the ureters. Okay. So from the calyxes to the pelvis, the area around here, and then finally to the ureter. which conveys it to the urinary bladder and to the urethra. So once again, you can look at the naming of the different parts. Here we have a little bit simplified diagram. Inside it, we have even the calyx is represented into the major and also the minor one and the major. So the minor ones are the ones that are still close to the pyramids and they are a bit narrower, but the major is a bit wider because it's a collection from various uh, pyramidal structures. And then it continues to the urethra. So let's talk about the renal artery. It's a blood vessel that carries oxygenated blood from the heart to the kidneys. Meanwhile, the renal vein, a blood vessel that carries the oxygenated blood from the kidneys to the heart. So what happens here is in our circulatory system, the heart pumps the blood, it comes through the renal artery, okay, it comes through the abdominal aorta, and then the renal artery is a branch of the abdominal aorta, and then blood runs through the kidney, and then in the due process it exits through the renal vein. So from the time of entrance of blood at the renal entry until the time it leaves at the renal vein, quite a number of events happen, and this forms the basic function of the kidney, the ultrafiltration and the selective reabsorption of materials that we shall be discussing basically here. So functions of the kidneys, one is to regulate the blood volume and electrolytes, regulate also the acid-base balance, excretion of nitrogenous waste, regulate blood pressure, and regulation of red blood cell production. Let's look at the condition called uremia. Uremia refers to the presence of blood of a substance that should be filtered into the urine. When the kidneys fail, these substances stay in the blood. Some of these substances are the nitrogenous waste, in the condition referred to as azotemia. Azotemia is the presence, the accumulation of nitrogenous waste in the blood, and this happens when the kidney fails to function to remove those waste. So these ones include elevated creatinine and urea levels. Measuring creatinine and urea levels, of course, is diagnostic for testing kidney diseases. And the kidney also secretes or respond to hormones that regulate blood pressure and red blood cells production. So, as we shall know, kidney function tests measures quite a lot, relies on the levels of urea and creatinine because we have the normal levels and we have the abnormal levels. So normally, what the laboratory does is to first look at these two parameters if we are trying to see if the kidney is functioning well, and if it is not filtering the nitrogenous waste, then you will find elevated levels of urea and creatinine, which can be toxic to the body. Let's look at the nephron, which we say is the functional unit of the kidney. Details. The nephron is composed of two parts. We have the renal tubules and then the renal blood vessels. The nephron unit is the one that produces the urine. So let's look at the kidney as a whole. Okay, and then you can see the drawing here. We are trying to take a small portion of it and then we examine further with our microscope because they are really microscopic, and this is what we are going to see. We are trying to visualize the functional unit of the kidney to give us a more better picture, and that's why we can see the diagram here. So in each of these parts of the kidney, this is what we see as the functional units. And this is how the blood vessels interact and also the filtration system, and then how they're also connected to the pelvis of the kidneys and then to the collecting duct, and then it goes down to the urinary bladder. So uh, the arrows here are trying to tell us the direction of movement of blood. Okay, the The one in red is for the arteries, and then the one in blue is the vein, which are going in the reverse direction. So blood goes and then blood leaves. So during the ultrafiltration, as blood is moving through the kidneys, what happens is that we have the filtration unit of the kidney, which is called the glomerulus, okay, inside the nephron. So blood goes reaching the glomerulus, blood goes here under high pressure, where we see the arrow, and then down here, there's filtration pores. We have pores which allow certain amount of water and solutes and certain uh, filtrates that are, are able to enter through the basement membrane of the uh, glomerular of the glomerulus. And then there are those that do not enter through the filtration and then they continue to move in the blood. And then it continues, okay? And then we have the vasaracta where the arteries is like a loop of capillaries where arteries meet veins. We know that arteries meet veins through a system of capillaries. So non-filtered elements are considered important elements that the body still needs, and then they go back into the circulation, and then they are circulated again. But then those that are unwanted will always be filtered from here. In a normal kidney, this will occur as simple as it is. But when there is a damage, then we start to see elements that are not supposed to appear in urine start to appear in urine. For example, if you see blood in urine, uh, that is already a problem because the red blood cells are too big. They are too big to pass through the basement membrane of the glomerulus. They are not supposed to enter through, but in a situation of kidney disease, I mean kidney damage, you will see that there is red blood cells, hematuria in urine. Okay, even proteins. Proteins are too big; they are supposed to pass through this. So when we see proteins in urine of certain amounts, it's telling us something is already wrong with the urinary system. So the next slide, we can look through this, is basically trying to describe the functions of the different parts of the nephron. Okay, the nephron part here we have the renal capsule, and then here is trying to give us the function. Within the capsule, I mean the corpuscle itself, we have the glomerulus and even the Bowman's capsule. So the glomerulus is the one that filters the blood to form what we call the ultrafiltrate. The ultrafiltrate is the one that comes out, what that has been filtered out containing those waste products. Uh, the Bowman's capsule collects and encapsulates the filtered fluid. A proximal completed tubule, this one has the role of the reabsorption of glucose, amino acids, and most of the filtered ions like sodium and chloride. So what happens is like this, when some of the fluid is filtered, it is not that everything that passes through the basement membrane of the glomerulus is unwanted. Some of them are wanted still, and then later on the kidney, I mean the proximal completed tubule will, will reabsorb them through the process of either selective reabsorption or just by passive osmosis. Okay, they'll be reabsorbed. So this is the function of the proximal completed tubule, and mostly those are the ions and sodium chloride, including many others. Now let's look at uh, at the the proximal, okay, the proximal tubule as well. PCT here means proximal completed tubule. It is still a continuation of this one. It is measurable. Its measurable is the absorption of water. So a lot of water can be filtered out, but to keep homeostatic balance, it can still reabsorb some of the water. And also secretion of waste products like drugs and toxins into the tubular fluid. So this is one of the parts where chemical or nitrogenous compounds or even drugs are also excreted. We know that when we take drugs, it is normally the kidney that deals with them in the excretory process. So some of this start to happen at the distal part of the proximal completed tube. The loop of Henle is the U-shape structure we saw in the previous slide. Uh, its function is further reabsorption of water and other ions like sodium and chloride. Okay. And then number two is establishment of an osmotic gradient in the renal medulla. So it keeps a balance through an osmotic gradient. We know osmosis being the process of movement of water from area of uh, low concentration, not just water, but even ions from area of low concentration to high concentration. So this balance is also maintained here in this part of the lobe of canal. We have the DCT, is a distal convoluted tubule. This is for fine-tuning of electrolyte and pH balance. Basically, this is for pH balance and electrolyte. That means in the distal, in the proximal convoluted tubule, we have seen some electrolyte being reabsorbed, but in the distal, this is like final adjustment. Okay, we are trying to see is the amount reabsorbed adequate or is it too much that others need to be excreted? So this one is like a final check to keep more of the balance in the absorption and reabsorption of certain electrolytes. But more importantly is the regulation of hydrogen ions to keep the pH balance. Number two, regulation of sodium, potassium, and calcium levels is part of the ions we're talking about. We have the collecting duct. This one further absorbs water and electrolytes. Concentration of or dilution of urine depends on the body's needs, and then collecting and transporting urine towards the renal pelvis. So this collecting duct plays one of the final important balance here. If the body needs quite a lot of water, then part of it can be reabsorbed. Okay, like during cold weathers where maybe you have taken a number of drinks, but the weather is very cold, you are not sweating. Okay, this collecting that will do the opposite because your body has retained a lot of water. So instead, what it will do is to make sure that a lot of water also uh, goes into the urine in the body, and then you will find yourself urinating so often on uh, those particular days than on dry days when there's too much sweating, where some of the body water is lost to other forms like the sweating. Okay, the nephron unit, we are looking at the blood vessels. The tubular and the vascular structures interact at the glomerulus within the Bowman's capsule and within the membrane of the tubular capillaries that surround the tubular network. Water and dissolved salts exchange between the tubules and the vasculature. We have three basic steps in the urine formation. One is what we call the glomerular filtration, which I've already mentioned, ultrafiltration. Two is the tubular reabsorption that happens in the proximal and the distal complete tubule, and also within the loop of canal. The loop of canal is the U-tube structure between the PCT, proximal convoluted tubule, and the DCT, the distal convoluted tubule uh, of the nephron. And then we have the tubular secretion. This is occurs within the tube. Okay, some solutes are actively secreted. All this is just to keep the balance between the osmotic pressure. Urine formation, we are looking at the glomerular filtration. So water and dissolved salts filters across the glomerulus into the Bowman's capsule. And then the glomerular filtration occurs when the pressure on one side of the membrane is greater than the pressure on the opposite side. Of course, at the afferent arterioles where the blood is coming in, the pressure is always higher than the side where it's exiting. So there will be that ultrafiltration. The following substances are filtered within the Bowman's capsule. We have the water, sodium, potassium chloride, glucose, uric acid. These ones pass easily. But large blood cells like proteins and even red blood cells, as already previously mentioned, doesn't pass through the pores. If glomerular pores are enlarged, they allow the filtration of proteins such as albumin, causing albuminuria, presence of albumin in urine. Normal urine is called ultrafiltered because it should contain no protein. The appearance of protein in the urine indicates glomerular dysfunction. Not all blood entering the glomerulus is filtered. Whatever is not filtered is via the afferent arteriole, which then extends as the peritubular capillaries. In glomerular filtration, water and solid are moving from the blood, that is the glomerulus, into the tubules. So we look at the glomerular filtration rate. We say it is the rate at which water and solid are filtered. So we can actually measure it. The rate at which water is filtered through the glomerulus. We look at what volume of water is passed through the glomerulus in what particular period of time. The blood pressure also determines the glomerular filtration rate. Normal glomerular filtration rate in the body is normally is normally in an adult is 180 liters of water for 24 hours. Okay, and then it is an equivalent of 125 ml of water per minute. Blood pressure determines the glomerular filtration rate, and if blood pressure becomes very low, like somebody in a stage of shock with systolic blood pressure of 60 and diastolic of systolic of 80 and diastolic of 90, and then that means the blood flow to the kidney and the glomerular filtration rate and even the urine output will all decline. So this is what happens when someone is in shock. Okay. Um, so a person with a urinary output of less than 400 ml for 24 hours is considered to be oliguric. Oliguric means little amount of urine or scanty. A continuing drop of glomerular filtration rate indicates that a person is developing renal failure. If a patient's blood pressure is declining, why should the urinary output be monitored? This is just a question. But we already mentioned that oliguria is a symptom of shock, and prolonged oliguria can cause further renal damage, such as acute tubular necrosis. So what is important is that we need to be to always monitor, because this might be an indication of a start of a renal failure, but we are so much worried when it becomes when it reaches the stage of acute tubular necrosis. The tubules, once they become necrotic, means when the tubules die, it is non-regenerative, and this can lead to end-stage renal failure. Urine formation, tubular reabsorption. So here we are looking at what is reabsorbed. Tubular reabsorption is a process of reclaiming water and solids from the tubular fluid and returning them to the blood. It involves passive and active transport mechanisms across the tubular epithelium. The main factors that determine the rate and extent of tubular reabsorption, one is the concentration gradient between the tubular fluid and the peritubular capillaries. The next one is the permeability of the tubular epithelium to the different substance, the solute, and the availability of transport proteins and energy sources for active transport, and then the influence of hormones and autoregulation on the renal blood flow and filtration rate. So we shall see that certain hormones are important for all these regulations to happen. Uh, we already said this, the amount of urine filtered in 24 hours, but however, a normal adult normally urinates 1.5 liters in 24 hours. Okay, on average, this is the amount of urine that an adult passes within optimal conditions within 24 hours. So the tube will reabsorb about 178.5 liters within 24 hours. So much of this water filter is actually reabsorbed uh, by, by the body. We are talking of 180 liters, but here we are only passing 1.5. So what is reabsorbed? 99% of the sodium that passes through the glomerulus is reabsorbed back because the body needs it. The same to water, almost more than 99%. Glucose is actually 100%. Okay, and we shall talk about this when we shall be discussing about what happens in diabetes, why they have glucose in their urine. Actually, 100 of it is supposed to be reabsorbed. Urea is 50% and creatinine is 0%. 0% meaning we don't reabsorb it. Okay? And 0% meaning we don't need to reabsorb this. just go to the urine. Let's look at this diagram. You can check your time and study it properly. It's trying. We have we have all. It's a summary of what we have discussed, but it's trying to tell us which particular solutes uh, passed out or are absorbed at which particular point of the kidney. So if we have, this is our renal cortex. Okay, we are looking at the kidney reabsorption and the secretion. Okay. Uh, the arrow in red is telling us the direction of flow and it's also telling us the filtration. So what goes in that filtration? We have water, salts, hydrogen carbonate, also hydrogen ion, urea, glucose, amino acids. Okay, these are smaller units of building blocks of proteins and then certain drugs. Reabsorption and secretion. So this arrow here is telling us active transport, the process by which these particular ions are regulated. While passive transport, so active transport involves use of some energy forms. This occurs against concentration gradient, but passive transport is just uh, along the concentration gradient. Could be with the passive osmosis process. So if this is the renal cortex, okay, we have our glomerulus here. We have the afferent arteriole. The afferent arteriole is the one, the branch of the artery that comes from the renal artery. Okay, and then blood is flowing through it under high pressure, and then it is filtered and it leaps through the afferent. So this part here is an artery. This one is also an artery. Don't confuse it as a vein. Okay, the veins are elsewhere. Here it is an artery. The purpose of this is just to perform the specialized function of filtering, okay, within the Bowman's capsule. So the efferent now goes, and this one continues now to reach the veins later. I mean through the capillaries, the basal capillaries, and then to the veins, and then blood moves in that order. And then, so what happens here is now our filtered fluid containing all these things that we have talked about. Okay. Now this one will move through this capsule, the tubule, and then it goes to our first proximal convoluted tubule. As this filtered fluid is going, already at different levels, different ions are leaving. We have potassium. Most of the potassium here uh, is getting out through passive transport. Okay, just also water, some hydrogen carbonate, and then this one continues. Here also there's an active. So where the arrow points is telling us where it is going. So here some hydrogen is absorbed into the tube, including also some kind of this is a nitrogenous waste. This is ammonia. Some of them is still taken in. Mixing here, sodium chloride and some nutrients are given out, and then it continues. Reaching here, more of the water is reabsorbed. This arrow is telling us it's reabsorbed into the body. What is inside this tube is going out. Okay, but what is going from this tube out is still being retained by the body. This is how you can make sense of this directions of movement and these arrows. So here it is continuing within the U tube here, the loop of Henle, which is U shape. As it is ascending, the ascending loop of Henle, you see here that most of this sodium chloride is reabsorbed. Okay, is reabsorbed, and we shall also see that some hormonal responses are involved in the homeostasis of this sodium balance, and we shall talk about those in the coming slides. What determines how much amount of sodium should be retained back into the blood, or how much should be allowed to go into the urine? Is what we shall talk about. So mixing here still more sodium chloride, okay, is also being reabsorbed in the system. And then we call this the thick segment of the ascending limb. And then our ultrafiltrate is continuing, it goes, reaching the distal convoluted tubule. We have more ions also reabsorbed, sodium chloride and then hydrogen carbonate ions. Here we also have potassium and also calcium, hydrogen ions being taken in. And then some part of water is also reabsorbed here, but not much as the one here. And then this continues until it reaches the collecting duct. We can still see how that some sodium still is even being reabsorbed. And then even some urea still might be reabsorbed and some water. Okay, and then this one goes down to our urinary bladder. Now, let's look at diuretics. This is already an aspect of pharmacology within physiology or anatomy. So we are looking at the role of the diuretics in the balance of these uh, processes in the kidney. So we said diuretics, these are drugs, they are used to increase the urine flow. Most diuretics block the reabsorption of sodium in various ways. That's blocking the reabsorption of water as well, because water follows sodium because of the osmotic gradient. A normal glycemic person filters glucose but reabsorbs all of it. Okay, as I mentioned earlier. But in hyperglycemic person, in the case of our diabetic patient, okay, uh, they filter a greater amount of glucose but cannot reabsorb 100% of it. So as a result, they are said to be glycospheric, and that's why the urine of a diabetic patient contains significant amounts of glucose. You might be wondering, uh, in, in the history of diabetics, diagnosis of diabetics, you might have heard that actually uh, initially at the start, people used to test the urine of a diabetic person and it was found to contain sugar. So this was an initial but also an important step towards the, the understanding of this particular disease. So tubular secretion, we say tubular secretion is the third and the last step in the urine formation. It moves very small amounts of selected items from the tubular capillaries into the tubules. The secret substance here are the potassium, hydrogen ions, uric acid, ammonium ions, and drugs. Tubular secretion normalizes the ionic concentrations as well as secretes substances such as uric acid and drugs. The secretion of hydrogen ions is usually accompanied by the reabsorption of the hydrogen carbonate, allows the kidneys to help regulate acid-base balance. We look at the aldosterone and the urine formation. So the aldosterone is a hormone, okay, and this one works on the distal tubule and upper collecting ducts of the nephron unit. So as I mentioned earlier, it is the influence of this hormone that helps the body maintain the homeostasis as regard this particular ions we are talking about. So it regulates if too much salt is needed to be retained in the body or too much of it need to be allowed to go into the urine. So it is sometimes referred to as the salt retaining hormone. Its effects are it increases the sodium and water reabsorption. So if our body is taking back a lot of sodium, that means yes, we are retaining uh, we are retaining a lot of this. And then what happens when more water and sodium is retained, meaning we are increasing the volume. Okay, so the blood pressure is normally raised, and that's why in renal hypertension you find that yeah, a lot of water is retained in the body and sodium and all those, and then the person develops high blood pressure just from renal origin. Okay, of course, blood pressure is increased. Uh, let's look at the tubular secretion. The distal tube is the site of aldosterone's action, the DCT. Okay. Why do you use a diuretic that blocks aldosterone? This is just like a brainstorming question. So when we use an aldosterone, I mean when aldosterone is blocked, we have sodium and water, they are not reabsorbed. Remember, we mentioned that the function of the aldosterone hormone is to make sure that more water and sodium is reabsorbed in the body. But when we use a drug which is diuretic, diuretics act by the principle of blocking the actions of this hormone, aldosterone. So you give somebody diuretic, we have the loop diuretics and both the thiazide diuretics, okay, pyrosemide, hydrochlorothiazide, the thiazide diuretic, all those works in a principle that it blocks the actions of this particular hormone. So when this happens, no reabsorption happens. Most of the sodium and water is passed into the urine. Okay. What happens is that okay, well, sodium, potassium will be retained. So we might have high levels of potassium in the body, but we will have already lost a lot of water and sodium, and then therefore the blood pressure goes down because the hydrostatic volume is reduced. In acute adrenal cortical insufficiency, why is the patient hyperkalemic with a soft and low blood pressure? So the answer is that the lack of aldosterone from the adrenal cortex prevents the absorption of sodium and water, thereby lowering the blood volume and the blood pressure. And then, in this case, the potassium is also excreted in the urine. Let's look at the renin-angiotensin-aldosterone system. Okay, so this system enables us to understand how these different organs interact in controlling the blood pressure. And this excuse me, sorry for that small interruption. So we said we are looking at the renin-angiotensin-aldosterone system. It enables us to understand how the different parts, the different organs interact to control the blood pressure, and later on it will also even help us understand how the diuretics work, how those particular drugs work, and even the different organ systems in regulation of blood pressure. So let's look at the liver. Starting with the liver, what we have here is our blood vessel. The liver produces a hormone which we call angiotensinogen. This is a prohormone. Angiotensinogen, and this one has an impact on the blood vessels. Okay, but this is a prohormone. It's produced in an inactive form. Okay, waiting for another activation for it to be active, to function. So the liver does this work to produce the angiotensinogen, and then the the juxtaglomerular cells in the kidney produce a common mix we call renin. Okay, this renin, its work is now to activate the prohormone, the inactive form of the angiotensinogen, into what we call angiotensin 1. And then this angiotensin 1 needs also another hormone that is produced from the lungs, okay, which is called angiotensin converting enzyme, SE. Okay, and then this angiotensin converting enzyme acts on angiotensin 1 and converts angiotensin 1 into angiotensin 2. Again, at this stage, we have the adrenal cortex. The adrenal cortex is a small rectangular shaped organ that always sits on top of the kidney. Okay, you might have seen this in your anatomy. This organ here is very important. It produces a lot of quite a number of hormones that are important in the homeostasis of regulation of salt and water balance that we might talk about. So the adrenal cortex produces another common, I mean, produce the aldosterone, okay, which which is which is acted upon, which stimulates the kidney either to increase the reabsorption or to increase the absorption of those particular solutes. So the end result is that it has an impact. Angiotensin 2 has an impact. It stimulates the adrenal cortex, and then the adrenal cortex releases aldosterone. Then aldosterone has an impact on the kidney to control salt and water balance. But at the same time, also angiotensin 2 also acts on the arterioles, causing vasoconstriction. And when vasoconstriction happens, uh, what happens to our blood volume? So you'll see how important really these these organs interact together in the maintenance of the blood pressure. So in the state that somebody has a liver problem, it means that already perhaps if the synthetic function of the liver is affected, we might not be having enough of this prohormone, you see, and then our homeostatic balance is affected because not enough of this produced, and then renin cannot be able to, even if the kidney produce the renin, not so much angiotensinogen is present to act on this. So this axis describes how like various organs interact, and understanding of it makes us understand how the blood pressure is regulated through the homeostatic inputs of all these particular organs. So activation of the renin-angiotensin aldosterone system releases aldosterone to the blood. The most common trigger activating the system are low blood pressure, diminished blood volume. Most important stimuli for the release of aldosterone is renin. Renin is an enzyme that activates the renin-angiotensin aldosterone system in a series of events, and it is released from the kidney in response to low blood pressure. Angiotensinogen is the inactive form circulating in the blood. I already mentioned this. Angiotensinogen 1 is released by the lungs. Angiotensinogen 2 is aldosterone released by the renal cortex, and then distal tubule kidney reabsorbs water, sodium, and excretes potassium. So why would a drug that blocks the converting enzyme lower blood pressure? These drugs are called the ACE inhibitors, the angiotensin converting enzyme inhibitors. They block the release of aldosterone and promotion of angiotensin 2, both of which elevate blood pressure. So for some of you who might have already known, this is the mechanism of action of the ACE inhibitors. Okay, the the captopril, eosinophil, all of them act by this particular mechanism. This is the point at which they act in trying to regulate blood pressure. Antidiuretic hormone (ADH) is released by the posterior pituitary in response to low blood volume or high concentrations. Water moves out of the collecting duct into tissue spaces and then into ventricular capillaries. Antidiuretic hormone decreases the excretion of water and causes excretion of concentrated urine. ADH also works primarily on the collecting duct of the nephron unit by determining its permeability to water. So this is what the ADH hormone does. The level of its axon is at the collecting duct. Okay, the collecting duct. Then it determines, it makes it more permeable to water, and then more the water is allowed into the collecting duct. Because ADH controls the permeability of the collecting duct to water, the presence of ADH, also called diabetes insipidus, prevents water from leaving the collecting duct and allowing excess water in the urine, called polyuria. This will cause hypovolemia. Excess ADH often seen in traumatic head injuries causes hypovolemia. We have what we call the natriuretic peptides. More specifically, I'm talking about the beta natriuretic peptide, which is always common in the diagnosis of heart failure. Uh, this peptide causes excretion of sodium, and it is secreted by the walls of the atrium in the heart, okay, in response to increase in blood volume. So when blood volume becomes massive, the walls of the atria in the heart respond by secreting this hormone because it is sensitive. It's trying to trigger a response to lower the blood volume. When the heart is failing, a lot of the blood remains in the heart because it's the weak. It's not able to push the blood through the circulatory system up the peripheral organs. So this becomes a homeostatic response by the heart muscles. The effects, I mean, it is this BNP is secreted by the walls of the ventricles in response to elevated ventricular volume. The effects of the natriuretic peptide are opposite of the effects of the aldosterone. So it's like a counteracting mechanism to ADH, to antidiuretic hormone. Since ventricular blood volume increases in the failing heart, Beta-natric peptide is used diagnostically in the assessment of heart failure. So if we take a blood sample, okay, someone has brought and is having signs of heart failure, and then we take a blood sample to test for the amounts of the beta-natric peptides, always we have the limit, the upper and the lower limits of normal. So if we find that BNP is increased beyond the normal limits, it's already telling us that there is already an element of heart failure in this particular person. Parathyroid hormone and calcitonin, these are also two important hormones that produce and they play a major role in the regulation of electrolyte and also blood pressure. Uh, this hormone is responsible for regulating calcium and phosphate. The effect, the phosphaturic effect, is the primary stimulus is low plasma level of calcium. Parathyroid hormone stimulates the renal tubules to absorb calcium and excrete the phosphate. Okay, so this is what it does. The hormone does. It is produced, stimulate the renal tubules to absorb a lot of calcium and pass out a lot of phosphate into the urine. The excretion of phosphate is called phosphaturic effect of parathyroid hormone. Also responding to plasma calcium levels is calcitonin secreted by the thyroid gland. Calcitonin is released in response to increased plasma calcitonin and causes the kidneys to excrete calcium and phosphate. Characteristics of urine, average, volume average, we already mentioned about this in 24 hours. Liguria is when we have a little volume of urine, and normally we say less than 400 ml in 24 hours. This is really small. Okay, and it might happen for various reasons. Maybe you are dehydrated, maybe you have not taken water for quite long, or maybe there is too much excessive sweating and you are exposed to kind of extreme body water loss. But also it can already be an indication of really kidney failure. There's no longer any filtration going as normal as you have seen in the normal person. Polyuria is the opposite. Okay, here we have too much volume for 24 hours, and this might happen in the diabetics, where the osmotic gradient is altered, where the more of the water is following the glucose outside into the urine. pH average, urine pH average is said to be as slightly acidic, okay? It's like 6.0. When you go uh, to the laboratory and then they test your urine, they are looking at parameters that will range within here, within 5 to 8. Acidic urine has an implication. Alkaline urine also has an implication. It tell us something is not right. It points to specific causes of parameters that alters urine pH in the various ranges. Parathyroid hormone and calcitonin. There's what we call specific gravity. Also, during urine analysis, we measure a specific gravity. Uh, we said sometimes the specific gravity of urine is slightly heavier than water. Okay, and it ranges from 1.001 to 1.0035, but sometimes can be more than that. And that's why you see in urine test, specific gravity is one of the parameters we use. We also look at the color, can be amber or straw colored, deep yellow in dehydration, or pale yellow in overhydration. So when you have taken a lot of water or you have taken a lot of fluid, you'll see that your urine normally will change. Okay, it will become pale. But if you have stayed working in the strong sun and you have not taken water and all this, you also have small volume of water, of urine, and even the color will be deep yellow. Okay. So why does the urinary high specific gravity, why does the urinary high specific gravity indicate dehydration? So the specific gravity is a ratio of the solid components in the urine to the volume. So it will tell us your status of hydration. Are you dehydrated or you are within the normal or you are overhydrated? So in dehydration, the volume decreases and the specific gravity will increases. The number of solute within the urine will be high because you are dehydrated. So little volume of water as compared to the solute. So this is what This is the relevance of a specific gravity in determining hydration status. Urinary retention and renal suppression. In urinary retention, the kidneys produce urine, but the bladder doesn't empty. In renal suppression, the kidneys do not produce urine. Okay, so mark the differences. Urine retention is often caused by anticholinergic drugs such as atropine, often used as a perioperative medication. Uh, let's look at what we call renal dialysis. We talked about uremia earlier. We said uremia is the presence of urine in the blood resulting from renal failure. So, if our kidney has failed, uh, the toxic elements in urine are no longer filtered and then they start to accumulate in the blood. This is the condition we're talking about, uremia. And it might be toxic to the whole body system, and obviously, we need to find a way to remove it. Okay, and that brings in the indication of dialysis. Dialysis removes the waste from the blood. In artificial kidney or the hemodialysis, waste and water diffuse out of the tube containing the patient's blood into a dialysate, which is a commercially prepared solution of water and electrolyte that is similar to the body's water and electrolyte content. So what happened here is that the kidney has failed. Blood is circulating all over the body, reaching the kidney, reaching the nephron, reaching the glomerulus and the basement membrane, but nothing is being filtered out. And then a lot of these waste products start to accumulate in our blood. Normally, what happens is that we can do, like you can see in this picture here, we can do the dialysis where we can insert an arteriovenous fistula. It's an part of the device is attached to an artery, another one is attached to a vein. So we know that as blood is passing through this machine is well calibrated in such a way that it is able to function like the kidney. Okay, and then as blood comes to the arteries, it's filtered and then returns to the vein. And then it removes all those nitrogenous waste, and this is what will at least keep our blood pure for this. And it's a very expensive procedure many times. Peritoneal dialysis. In peritoneal dialysis, the dialysate is run into the patient's abdominal cavity, and the patient's peritoneal membrane acts as a dialysing membrane. After some time, the dialysate containing the waste is removed. Dialysis takes time, often 4 hours every third day, so it can be disruptive to patients' lives. Let's look at our urinary bladder. Okay, so we have the ureters coming from the kidney. Down here we have the prostate gland. This is a man for this case. And then we have the pelvic floor muscles, and then this is the urethra. Okay, so from urethra, the urinary bladder, and then the urethra. Uh, Basically the same thing here. Okay, you can just appreciate these anatomical diagrams. Here we are looking at the location from the urinary bladder from a side view. Okay, so we have the male here, and we have the female. You can see here the uterus and yeah, the bowel behind the rectum and all those. Uh, the bladder is the trigonal bladder is formed by the entrance of the two ureters and the exit of the urethra. The detrusor muscle is the smooth muscle that causes bladder contraction and relaxation for expelling urine. The detrusor muscle forms the internal sphincter of the bladder. The bladder's external sphincter is composed of skeletal muscle, which is under voluntary control, but also there is we have the smooth muscle. Okay. So how could a urologist access a stone in the right urethra? A tube could be inserted into the urethra, bladder, and the right ureter, and a grasping device can then extract the stone. This is just trying to tell us some practical procedure that we can do. Sometimes we get what we call um, uh, uh, the stones. You know, kidney stones can lodge in the urinary in the ureter. So this is one of the procedures that can be done to expel them. Micturition, this is the urination reflex. So what happens during urination is that the bladder fills and stimulates the stretch receptor. The urinary bladder has receptors that are sensitive to stretch. So as volume is increasing, the receptors triggers a signal. Okay, sensory signals are sent to the spinal cord, and then the motor signal feedback is sent to the bladder. And when this motor feedback is sent, then there's a contraction of the bladder. When the bladder contracts, the internal sphincters relax. Okay, and urine comes out. Whereas the internal sphincter is voluntary and relaxes as part of the micturition reflex, the external sphincter is composed of skeletal muscle and is under voluntary control, allowing you to hold it. So what we are trying to see here is that the inner smooth muscle of the urinary bladder is under involuntary control. You cannot control it. It's actually controlled by this particular reflex. It's involuntary. But the extrinsic muscle, the external sphincter is composed of skeletal muscle. It's a skeletal muscle, so that means you have the power to control urine. Okay, you can control urine for some time because you can decide that it is under voluntary control. During micturition, the deep muscle contracts and the two sphincters relax. What urinary structure is brought under control during 40 training of a toddler? This is a question. The external sphincter is brought under control. Here we are trying to exercise, we are trying to train this child to exercise the external sphincter because it's under voluntary control so that the side gets used to, okay, and prevent bedwetting. The urethra is a tube that carries urine from the bladder and to the outside. The muscular layer of the urethra contracts and expels urine during nutrition. What is one way the male and female urethra differ? In the female, the urethra is shorter. It's 1.5 inches. In the male, is a bit long, an average of 8 inches. This is an average. It might not be a clear cut, but of course, it's a range. Autonomic movement due to some muscles and internal sphincters have muscarinic receptors, the muscarinic agonist effects, and then also we have the anticholinergic antidepressants effects. So certain uh, muscarinic agonist affects the function of the sphincter, and also the anticholinergic antidepressant drugs also have effects on the depressor muscle. Autonomic stimulation, we said activation of the muscarinic receptors causes the depressor muscles to contract and the sphincter to relax. This action causes the bladder to expel the urine. This can lead to two pharmacological issues. First, if a patient is experiencing urinary retention, he or she may be given a muscarinic agonist such as botanical or urecoline, the other name. And persons who take preoperative drugs such as atropine and those who take anticholinergic antidepressants. So what happens is if the autonomic function is impaired and if the patient experiences urine retention, we say we give a muscarinic agonist. Agonist means something that supports the action of muscarinic agents, and the drug that does that is the botanical. Okay? And when we give this drug, normally it should be able to relieve the patient to be able to pass urine. Okay, we have come to the end of this lecture. Thank you so much for being with me and see you in the next lecture. Bye bye.